Hi everyone, I'm Alex Wu, and the title of my talk is Bayesian Information Sharing Enhanced Detection of Regulatory Associations in Rare Cell Types. The work I'll be presenting is joint work with Zhen Peng, Bonnie Berger, and Hoon Cho. Gene regulation involves the intricate coordination of many biomolecules. Differences in the coordination of these biomolecules contribute to the diversity of cellular functions and phenotypes observed across different cell types and cell states. Over the past several years, single-cell sequencing technologies have allowed us to characterize many cell types and cell states that were previously obscured when sequencing in bulk. Rather than profiling cells across an entire population of cells and simply observing an average of a population of cells, single-cell methods allow us to measure a particular genomic feature for each cell in a population. As a result, not only can we better identify cell types and cell states with these technologies, but they give us an opportunity to study the gene regulatory patterns that underlie these cell types and cell states. How do we characterize the gene regulatory associations that underlie these different cell types and cell states? One straightforward approach to inferring gene regulatory associations for a cell type in a single-cell RNA-seq dataset is to take the cells that belong to that cell type and apply a gene regulatory network inference algorithm on just this subpopulation of cells. While this approach may be effective for cell types that are represented by a large number of cells, for rare cell types, which often are of a great deal of interest in single-cell studies, the gene regulatory network inference task may be underpowered due to the small sample sizes inherent to these rare cell types. This is the primary challenge that we hope to address. The task of inferring gene regulatory associations for rare cell types or rare cell states is often inherently underpowered. We can see this by inspecting the edge scores for networks inferred across various cell types in a single-cell RNA-seq dataset, profiling the mouse blood lineage. In this dataset, 24 cell types have been detected. The cell type with the largest number of cells consists of 1,860 cells, while the rarest cell type consists of only 30 cells. We considered three different network inference approaches: Pearson correlation, Gini-3, and PIDC. Pearson correlation is a common metric for calculating co-expression in single-cell studies. Gini-3 is a random forest regressor-based method, while PIDC is a mutual information-based approach. Gini-3 and PIDC are two of the top performing network inference algorithms in a recent single-cell network inference benchmarking study called Beeline. For each cell type in the dataset, we estimated the noisiness of the outputs of these network inference approaches by calculating the standard deviation of each edge's score when applying these network inference approaches on several bootstrap samples of cells for each cell type. In this box plot, we've plotted the standard deviations for the top 1% of edges for each cell type, ranking the cell types by the number of cells that make up each cell type. We can see from this plot that across the three different network inference algorithms, as the number of cells decrease for a given cell type, the standard deviation increases, indicating a greater degree of noisiness in the network estimates for cell types represented by small samples of cells. In order to overcome the challenge of gene regulatory network inference in small sample size settings, we developed a Bayesian information sharing framework called ShareNet. In this framework, we draw upon the intuition that many regulatory interactions and non-interactions are shared across different cell types due to shared developmental lineages, regulatory programs, or biophysical constraints. By propagating information across related cell types, we hope to reduce noise and boost the accuracy of inferred networks in all cell types. In our framework, we also allow for multiple cell type to cell type sharing patterns. This feature is important as a regulatory interaction may be globally shared across all cell types in a dataset, shared across only a few related cell types, or unique to a single cell type. ShareNet automatically learns and leverages the specific sharing pattern that is most appropriate for each edge. ShareNet does this using a Bayesian framework, in which these sharing patterns and the network estimates outputted by a user's chosen network inference algorithm are governed by a hierarchical Bayesian model. As a summary of ShareNet and its usage, instead of taking the cells from each cell type and only applying a network inference algorithm for these cells, we take the networks inferred using this approach as a set of initial network estimates, which serve as input to ShareNet. ShareNet learns a set of adaptive sharing patterns that it uses to share information across cell types for each edge, which generates a set of enhanced network estimates for each cell type that accounts for the learned cell type relationships. In more detail, we represent the multiple cell type sharing patterns as a mixture of multivariate Gaussian distributions, with each mixture component corresponding to a unique sharing pattern. We then model the true scores for an edge across cell types as a draw from this mixture of multivariate Gaussians. For a dataset with C cell types and for K mixture components or sharing patterns, we represent this using the variables Uij and Zij, where the ij subscript corresponds to the edge connecting gene I to gene J. Uij is a categorical variable that represents a specific mixture component that corresponds to the edge ij. The variable Zij is then drawn from the multivariate Gaussian mixture component associated with Uij and is a vector that represents the true scores of the ij edge across the C cell types. The C by C covariance matrix for each Gaussian mixture component represents a unique cell type sharing pattern and captures the relationships between the true edge scores of the C cell types. A stronger sharing pattern between two cell types would be represented by higher correlations between the two cell types, while a weaker sharing pattern will be represented by weaker correlations between two cell types. As a brief example for this part of the model, let's consider 4 cell type specific networks and zoom in on the part of each of these networks that concern a single transcription factor, here in blue, and several putative target genes, represented here in orange. The weights of the lines between the transcription factor and each target gene for each cell type represents the true edge score. For a single edge ij between the transcription factor and a target gene, we represent the scores for this edge across the 4 cell types as the vector Zij. These true scores are drawn from a specific mixture component of the Gaussian mixture model, where the relationship between these true edge scores is governed by a specific cell type sharing pattern. Going back to the model, the edge scores that we observe as outputs from a network inference algorithm are then modeled as noisy draws from univariate Gaussians centered at these true edge scores. Here, Eij superscript C is the observed edge score for edge ij for cell type C, and its model is a draw from a Gaussian centered at true edge score Zij superscript C. The degree of noisiness for each edge in each cell type is specified by the variance parameter of the univariate Gaussian, which we estimate using a bootstrapping approach. Lastly, in our model, we also include a normal Wisher prior on the parameters of each multivariate Gaussian mixture component for regularization. Because the Zij latent variable represents the true edge scores for edge ij across the C cell types in a dataset, we're interested in calculating the posterior density of the latent variables in the Bayesian model given the input data. Specifically, we are interested in using the mean of the posterior distribution for Zij as enhanced edge scores that account for the sharing structure implied in our Bayesian model. Exact inference of the posterior distribution is intractable for our model, so we leverage variational inference to approximate the posterior. We applied ShareNet first to a set of single-cell RNA-seq datasets from Beeline, a single-cell RNA-seq network inference benchmarking study. Here we considered 5 cell types, and to account for small sample sizes, we took a population of dendritic cells and downsampled the population to 50, 100, or 200 cells, while keeping the population sizes of the remaining cell types the same. We then considered the effectiveness of using ShareNet to enhance the network estimates for the downsampled dendritic cell populations. We did this by comparing the inferred network for dendritic cells with 3 reference networks: a protein-protein interaction network curated from the string database, a non-cell type specific ChIP-seq network, and a cell type specific ChIP-seq network curated only from assaying dendritic cells. We tested the effectiveness of ShareNet across the 3 different network inference methods we mentioned earlier: Pearson correlation, Gini3, and PIDC. In this figure, the lighted shaded bars correspond to the AUPRC values of networks inferred without using ShareNet. We can see across all 3 network inference methods and across the various reference networks, the AUPRC values increase as more cells are considered, providing further evidence for noisiness of network estimates in small sample size settings. The darker shaded bars correspond to the AUPRC values when using ShareNet. As we can see in this plot, the darker shaded bars indicating the ShareNet inferred networks are generally higher than their counterparts inferred in the absence of ShareNet across the various downsampling scenarios, network inference methods, and reference networks. For 2 full-scale single-cell RNA-seq datasets, we also compared the accuracy of networks inferred with and without ShareNet. The first dataset we analyzed was a single-cell RNA-seq dataset from the Tabular Mirrors Consortium that contained an extensive collection of cell types isolated from 20 mouse organs and tissues. The second dataset profiled the mouse blood lineage and consisted of cells along a differentiation trajectory in early development. Similar to the Beeline dataset analyses, we compared the network for each cell type with a PPI network, a non-cell type specific ChIP-seq network, and its corresponding cell type specific ChIP-seq network. On the x-axis here, we have the accuracy of networks inferred without ShareNet. On the y-axis, we have the accuracy of networks inferred with ShareNet. Each point corresponds to a specific cell type's accuracy with respect to each of the 3 different reference networks, with the accuracy for each reference network represented by a different color. We see that most of the points in both the Tabular Mirrors and mouse blood lineage datasets, and across the 3 different network inference methods, lie above the diagonal, meaning that the accuracy of the networks inferred with ShareNet were generally higher than the networks inferred without ShareNet. P-values from a one-sided Wilcoxon sign rank test comparing the accuracy of networks inferred with and without ShareNet also provide support for ShareNet's ability to improve the accuracy of most cell types networks. ShareNet's ability to increase the accuracy of networks opens the door to a wide range of downstream analyses. Using the Tabula-Mears dataset, we applied ShareNet on Pearson correlation networks, which achieved the most accurate networks in the previous set of results. We first looked at the cell type sharing patterns that are automatically learned by ShareNet. Here's the correlation matrix associated with the highest mixture weight in our model. From this matrix, we can see that cell types that belong to the same broader cell type category in the Tabula-Mears dataset exhibit strong sharing patterns with each other, which suggests that ShareNet captures and uses cell type similarities that are biologically meaningful. For the Tabula-Mears dataset, we also were interested in evaluating if ShareNet allows us to more effectively compare networks of related cell types. The Tabula-Mears dataset includes mesenchymal stem cells isolated from the adipose tissue, trachea, limb muscle, and the diaphragm of mice. We wanted to look into the specific differences in networks between these four types of mesenchymal stem cells with different tissues of origin. First, we looked for the set of edges that are added or removed from networks when using ShareNet. Here, the y-axis corresponds to the significance of enrichment for a Go biological process term. The purple box plots represent the significance of the Go terms that are enriched in the edges added when using ShareNet, while the pink box plots represent the significance of Go terms that are enriched in edges that are removed. Across a range of thresholds for retaining top edges in each network, we see that the set of edges that are added when using ShareNet are consistently more strongly enriched for Go biological processes. Meanwhile, edges that are removed tend to have noticeably less functional enrichment. This set of results suggests that ShareNet omits edges that are more likely to be spurious, while at the same time adding edges that are more likely to correspond to functional regulatory interactions. We also compared the four different mesenchymal stem cell networks and identified the edges that were specific to each of these networks. We found that the cell type specific edges in each of these networks are significantly enriched for differentiation development pathways that correspond to their tissues of origin. We performed the same set of analyses for networks inferred without ShareNet and found that of the four tissues networks, only one of the networks, the network for the trachea, had unique edges that were significantly enriched for the relevant tissue-specific differentiation pathway. The differentiation pathways related to fat, the limb muscle, and the diaphragm were only significantly enriched when using the networks inferred using ShareNet. Lastly, we were interested in using ShareNet to study the rewiring of regulatory associations in dynamic biological processes. We used the mouse blood lineage dataset, which contains cells collected from whole mouse embryos from stages of early embryonic development. Here we used Gene3 with ShareNet since this combination yielded the strongest performance for the mouse blood lineage dataset from our earlier accuracy results. Like before, we see that the cell type sharing patterns automatically learned by ShareNet cohere with the expert annotated cell type labels, as well as the underlying developmental trajectory. Cell types that are annotated with the same general cell type label have stronger sharing patterns with each other, and cell types that are proximal to each other in the trajectory also tend to have stronger sharing patterns with each other. We also looked more closely at the path of 10 cell types, connecting mesodermal cells at the beginning of the developmental trajectory and erythroid cells at the end. In particular, we wanted to find edges that are stably activated or deactivated during the course of differentiation in order to detect important changes in network rewiring that contribute to changes in cellular identity. We define a stably added edge to be an edge that's absent in the earliest mesodermal cells, emerges in an intermediate cell type along the trajectory, and then is maintained until the last set of erythroid cells. We define stably removed edges in a similar manner, but these edges correspond to edges that are initially present in the earliest mesodermal cells and then are permanently removed in an intermediate cell type along the trajectory. We also search for edges that are unstable, which we define to be edges that change their on or off status more than 4 times over the course of the path of 10 cell types. The number of each of these types of edges is represented on the y-axis, and we performed searches for these edges across a range of thresholds for retaining edges by their scores, represented here on the x-axis. We found that the number of stably added and the number of stably removed edges is consistently higher in networks inferred using ShareNet compared to without ShareNet. Meanwhile, the number of unstable edges along the developmental trajectory was noticeably higher in networks inferred without ShareNet, which reflects the higher noise levels in this set of networks compared to the networks inferred using ShareNet. In summary, inferring gene regulatory network structures of specific cell types is an important next step in characterizing the diversity of cell types or cell states within a population of cells. we presented ShareNet to help combat the issue of small sample sizes associated with rare cell types or cell states. ShareNet serves as an information sharing framework that automatically learns a set of interpretable cell type to cell type sharing patterns that improve the accuracy of inferred networks. At present, we have only explored ShareNet within the context of inferring networks using single-cell RNA-seq data. With the rise of single-cell multimodal datasets that profile multiple genomic or epigenomic features simultaneously in the cell, we may now have the opportunity to ask the question of how we can extend ShareNet by leveraging different data modalities simultaneously to infer networks in small sample size settings. We hope to explore this question more in the future. This concludes my talk. I'd like to thank several people for their help in making this work possible. I'd like to first thank Hoon Cho and my advisor Bonnie Berger for their mentorship, guidance, and support throughout this project. I'd also like to thank Jan Pang for his help in providing crucial insights and discussions for this project. Lastly, I'd like to thank the members of the Berger Lab for their advice and support along the way. I'd be happy to take any questions.